Hello everyone, welcome to the first lecture of this course. So first let's try to get some introductory idea about the enzymes. So why enzymes are required to generate energy? Every living systems, okay, they need energy and they uh, make this energy from the environment. For example, we usually consume a substantial amount of sugars as our food, right? And from the sugars we get energy. How? When sugar, in presence of oxygen, sugar breaks down into carbon dioxide and water, and in this process release uh, many free energy, and we use this free energy to do our everyday work. Like we use this energy to think, to move, to taste, to see, and every other works that we do every day. Okay, but and this reaction is a very thermodynamically favorable, highly exergonic reactions. But yet, if you just keep sugar, like we all store sugar, keep sugar in our home, right? So if we just keep sugar open, this conversion will not happen rapidly, right? The sugar will not get, the sugar will not convert into carbon dioxide and H2O, right? But when we consume sugars, this conversion happens very rapidly. Okay, instantly after consuming sugars, our body break it down into carbon dioxide and water, and we get the energy to work. So, why it happens inside our body? It's not happening outside, but inside our body, it's happening very rapidly. Okay, so difference is in catalysis. Inside our body, catalysis occurs. Okay, and so, because of the catalysis, the breakdown of sugars is very rapid rapid there. Okay, so that means without catalysis, actually we will not be able to sustain. Okay, our life will be not sustainable. Okay, so that means catalysis is inevitable. So who do the catalysis inside the body of a living things? Here comes enzymes. Basically, enzymes are the biological catalyst. Okay, so they are the reaction catalysts of biological systems, which is the most remarkably and highly specialized proteins. Okay, so why enzymes are so special? Because enzymes have some extraordinary characteristics. Enzymes have extraordinary catalytic power, often far greater than that of synthetic or inorganic catalysts. Okay. They have a high degree of specificity for their substrate, very specific for their substrate. Okay, and they accelerate chemical reactions tremendously. They accelerate the rate of the reactions tremendously. Okay, speed it up, make it very fast, and they function in aqueous solution under very mild conditions of temperature and pH. Okay, the major liquid of our body is water, that is aqueous, and enzyme can function in this aqueous solution very efficiently under very mild conditions of temperature and pH. Right? If enzyme would need high temperature or high pH, they will not work for us. Right? We human cannot survive in very high pH or high temperature. Okay. However, some non-biological catalysts also have these properties. Enzymes are biological catalysts. Okay. So let's talk about a little bit about the history of enzyme. Okay. So when the idea of enzyme first came out in late 1700, uh, some scientists shows that meat can be digested by the secretions of the stomach. Stomach secretions can digest meat. Okay, and scientists keep continued their research, and in 1800s, with examinations of the conversion, researchers found that starch can be converted into sugar by saliva or using various plant extracts. Till now, people actually didn't know that who is exactly doing this conversion. Okay, just people just know that this kind of conversion is possible by using like stomach extract, saliva, and various plant extracts. The name enzyme was not coined yet. Okay. Later in 1850s, Louis Pasteur, he concluded that fermentation of sugar into alcohol by yeast is catalyzed by ferments. Okay, and from his observation, he postulated that these ferments were inseparable from the structure of the living yeast cells. Okay, I mean, he said that only the living yeast cell can do this conversion. I mean, can produce alcohol from the sugar. Okay, and this view, this is his view called vitalism, and this view prevailed for decades, for a long time. However, I mean, scientists they keep continue their research, and in 1897, a scientist named Edward Buchner, he discovered that not only the living yeasts, the yeast extracts could ferment sugar to alcohol. Okay, the living yeast is not required to convert sugar into alcohol. Even yeast extracts can do that. Okay, so his observation provide that fermentation was promoted by molecules that continued to function when removed from the cells. Okay, and Buchner's experiment at once marked the end of vitalistic notions. Okay, and the dawn of the science of biochemistry. And the science of biochemistry literally started from there. And then a scientist named Frederick W. Kuhne, he gave the name enzymes. I mean, Frederick W. Kuhne, he first coined the word enzymes, okay, to the molecules detected by Boucher, okay, Bosman. Finally, in 1926, a scientist named James Summer, he was able to isolate and crystallize an enzyme which name is urease, and it is a breakthrough invention in the early enzyme studies. Okay, and Summer found that urease crystals consisted entirely of protein. I mean, he not only isolate and crystallize the urease, he also was able to find out that these urease crystals are entirely a form of protein or made of protein. And from his observation, he postulated that all enzymes are protein. Okay. However, as there was no other examples, his idea was controversial. I mean, not all the scientists or all the people accepted his idea. However, his idea, I mean James Summer idea, got very widely accepted around in 1930s. I mean, at that time, two scientists named John Northrop and Moses Kunitz, they crystallized various other enzymes like pepsin, trypsin, and also other digestive enzymes, and they also found that all those enzymes were proteins. Okay, so thus the Summers conclusion was widely accepted. Okay, and during this period, I mean in 19 first half of the 90s, a scientist named JBS Halden, he wrote a treatise titled Enzymes. Okay, so although the molecular nature of enzymes was not fully understood or appreciated at that time, but Halden made the remarkable suggestion that he made a remarkable suggestion that weak bonding interactions between enzyme and its substrate might be used to catalyze a reaction. Okay. So anyway, we're done with the history. Let's go back to enzyme. So we just heard that enzymes are proteins. So do all the enzymes are proteins? It's actually not true. You know, there are some RNA which can also act as enzymes. They are called ribozymes. So basically, I mean, all the enzymes are proteins except the ribozymes. Okay, and the number of protein enzymes are actually greater. Okay, actually most of the enzymes they are proteins. Okay, so if we want to understand enzymes, basically we need to understand proteins. Okay, so what are proteins? Proteins are the strings of amino acids folded up into distinct structures. Okay, and the functionality or properties of protein, that is enzymes, are largely determined by its three-dimensional structure of protein. Okay, So I know you all actually know this and you have learned this in other courses, yet just for recap. So you know that there are several levels of protein structures. How many? Usually, four. There are four levels of protein structures: primary structures, secondary structures, tertiary, and quaternary. So before we delve into enzymes, let's have some recap on the protein structure. Okay, so we'll start with protein structures. Okay, so the primary structure. What is primary structure? Okay, a primary structure is a sequence of chain of amino acids. Amino acids they get bound. Two amino acids they get bound through peptide bond formation. Okay, then another amino acids bind, amino acids bind, another amino acid binds, and they form a chain. And this chain is the primary structure of proteins. Okay, and then hydrogen bonding of the peptide backbone of the primary structure happens, and it causes uh, the secondary structure of the protein. Due to the formation of hydrogen bonding within the primary protein structures and some parallel primary structures, they cause some changes in the shape of the primary structures. Okay, we'll talk about this structure later in details later, and this becomes secondary protein structures. And then these secondary protein structures, some more interactions occurs within them, and they get folded. Okay, like this is one example. So this is called the tertiary protein structure. This tertiary protein structures is the functional structure of the proteins. When the amino proteins they get folded into tertiary structure, now they can become functional. They do the catalysis. Okay, and there is actually another level of protein structure which is called quaternary protein structures. So, and in the quaternary protein structures are made of two or more tertiary protein structure. Okay, so now let's talk a little bit more about this structure, the primary structure of proteins. Okay, covalent bonds, mainly peptide bonds linking amino acid residues in a polypeptide chain, the chain of amino acids. Okay, and the most important element of primary structure of protein is the sequence of amino acid residues, because the function of the final protein actually depends mainly on this amino acid sequences. Okay, and each protein has a distinctive number and sequence of amino acid residues. Okay, so this slide shows the peptide bonds. You already know this. I actually put it here just for your recap. Okay, so the peptide bond, it forms between the carboxyl group and amine group of the adjacent amino acids. Okay, so now secondary structure. So secondary structure is the local folded structures that form within a polypeptide chain due to interactions within adjacent amino acids. Okay, so the if the between the adjacent amino acids, interactions occur. Basically, these interactions are mainly hydrogen hydrogen bond interactions. Anyway, so particularly stable arrangements of amino acid residues giving rise to recurring structural proteins. Due to the interactions, the secondary structure of protein is formed, and they give some recurring structural patterns. There are three patterns: helicals, pleated sheets, and tars and these structures are largely stabilized. The secondary structure of proteins are largely stabilized by weak non-covalent interactions. Okay, so now I'll talk about this structural pattern of secondary protein. Okay, so first, alpha helix. So this figure shows the model diagram of the alpha helix secondary structure. So the polypeptide backbone, in case of alpha helix, the polypeptide backbone is tightly wound around an imaginary axis. So you can see this is an imaginary axis. So the polypeptide chains, they are tightly wound around this imaginary axis. And the R groups, this brown color one, you can see the R groups of the amino acid residues, they protrude outward, protrude outward from the helical backbone. This is the backbone, and the amino acids, they are protruded outward, right? And the repeating units is a single turn of the helix which extends about 5.4 angstrom along the long axis. So I mean every after 5.4 angstroms this repeats, this structure, I mean this unit repeats. Okay, so that was alpha helix, and then the beta sheet is a pattern of secondary structure. So beta sheet, in beta sheet the backbone of polypeptide chain is extended into a zigzag form. Alpha helix were helical around the imaginary axis, but in case of beta sheets they form a zigzag. They form a zigzag form. Okay, and arranged side by side to create a structure resembling a series of plates. And hydrogen bonds form between adjacent segments of polypeptide chains. Hydrogen bond. Okay, and the R groups of adjacent amino acids protrude from the zigzag structure in opposite direction. Okay, if one this one, another this one. Okay, opposite direction. And the adjacent polypeptide chains in a beta sheet can be either parallel, okay, either parallel or anti-parallel. Okay, so then tertiary structure. Basically, tertiary structure is the functional structure of the proteins or the enzymes. So, and tertiary structure, they describes all aspects of the three-dimensional folding of the polypeptide. They can be different types like fibrous, I mean the fibrous proteins or globular proteins. So, what is fibrous proteins? Polypeptide chains arranged in a long strands or sheets like this, long strands or sheet. Okay, and they usually consist largely of a single type of secondary structure. Okay, and their tertiary structure is relatively simpler. Okay, they're made of mainly one type of secondary structure, and their tertiary structure is relatively simple. And usually these proteins, they provide like support, shape, and a structure and external protection to vertebrates or vertebrates. Okay, like for example the skin. Our skin is mainly produced by fibrous protein. Our muscle cell. Okay, these are fibrous proteins. Okay, and then there is another type of tertiary structure which are called globular proteins. Okay, so globular proteins are polypeptide chains folded into a spherical or globular shape. That's why they are called globular shapes. Okay, and globular proteins often contain several types of secondary structures, whereas the fibrous proteins used to contain one type of secondary structures, but globular proteins, they contain several types of secondary structures. And who are the globular proteins? Most of the enzymes and regulatory proteins are globular proteins. Like for example, enzymes. Like our body has many different type of enzymes, right? Trypsin, chymotrypsin. These are globular proteins. Okay, so the most enzymes and regulatory proteins. So regulatory proteins, for example, hormone, say insulin. Insulin is a regulatory protein, right? It's not an enzyme, but it's a protein. So this type of regulatory proteins and enzymes, they are mainly globular proteins. Okay, because actually the due to their globular shape, it's easy for them to move, and the regulatory proteins and enzymes, they need to move throughout our body, right? Otherwise how they will do the function. Okay, but in contrast the fibrous protein, they basically they don't need to move, right? Suppose the protein in our skin, do they need to move to do their function? No. Okay. Anyway, the final structure, the quaternary structure of protein. Okay, so the quaternary structure of protein results from interactions between the subunits of multi-subunit proteins or large protein assemblies. Usually, at least two tertiary structure, at least two. Okay, I mean, so that means at least two or more tertiary structures of proteins, they interact with each other and form the quaternary structure. Okay, so that's why when a protein has two or more polypeptide subunits, they form quaternary structure, at least two. Okay, so protein quaternary structure ranges from simple dimers to large complex. I mean, a quaternary protein can have only two subunits, or they can have many subunits. Okay, so that was all about the introduction of enzymes. So in the next lecture, we'll go back to the enzyme. Okay, so we'll study more about the enzymes. Okay, see you then.